Thank you, Tim. Okay, so hello, dear members of the jury, professors, fellow students, and other attendants. I'm here to present you my master thesis, a meta-analysis on the effects of ocean acidification and warming on Arctic organisms. As most of us know, the atmospheric carbon dioxide concentrations have undergone an exponential increase since the start of the industrial era around 1750. Up until 1950, this was mostly due to deforestation and other land use changes. From 1950 onwards, burning of fossil fuels became the main source of emissions. Two major consequences of this have already become apparent, such as the 1 degree Celsius increase in temperature, but also there has been an estimated decline in the pH value of the world's oceans of around 0.1 units. In a process known as ocean acidification, which is depicted here in a simplified scheme. In short, CO2 gets absorbed by the oceans, where it reacts with water molecules to form carbonic acid, which then dissociates into protons and carbonate ions, thereby reducing pH and increasing acidity. A number of biological responses have already been observed, such as an increased energy expenditure by calcifying organisms for the maintenance of their calcium carbonate shells, but also behavioral responses have been recorded in fish, such as decreased predator avoidance, and for some autotrophs, even positive effects on the growth have been observed. Responses also seem to differ between life stages, with earlier life stages often being more sensitive, but even populations within a species tend to differ in their sensitivity based on their historical exposure to low pH conditions. It shows the Arctic Ocean as our study region because it is well known as being a bellwether for prospective changes. When it comes to temperature, for example, the average global temperature is expected to increase by around 3.7 degrees by 2100. Whereas the Arctic region is expected to warm by around 8.3 degrees Celsius. But also for acidification. Low temperatures, namely, increase the solubility of CO2 in the water, and increased precipitation and ice melt cause a dilution of total alkalinity, thereby further reducing pH values. The region is also known as experiencing the largest natural pH variability in surface waters, due to the extreme differences in primary productivity between winter and summer. Now for the objectives. We wanted to find out how and to what extent Arctic organisms are affected by both ocean warming and acidification. We wanted to see exactly which biological processes are affected in which way. And we also looked for sources of variability within these biological responses. As mentioned before, we did this through an analysis in which we compiled the results of a large number of individual studies coming from the primary literature. We found these articles in the Web of Science and Pangea databases using relevant keywords and filters. The list of articles that we obtained from this was then screened by title and abstract to make sure they were relevant to the questions we asked here. So this list of 134 articles was then, we then screened the methods section of each of these to make sure there were actually studies done in the Arctic region, and to make sure they showed a control and treatment value. So we then extracted 1,616 responses from these articles, for which we categorized all the data. And did an extensive screening based on, amongst others, the criteria stated here. In order to obtain a data set of comparable results and to minimize biases. After this, we ended up with a total of 167 responses coming from 42 articles with which we conducted the subsequent analysis. So each control and treatment value were transformed into a single natural log transformed response ratio, which should be interpreted as such. So a value of 0 would indicate there is no effect of the treatment. A positive value would indicate a positive effect, and a negative value, a negative effect. For each LNRR value, we also calculated an effect size variance, so that studies with higher sample size and or a low variation were given a higher precision. We then calculated the overall mean effect sizes and corresponding 95% confidence interval using weighted random effects models, so that studies with a higher precision weigh more heavily on the eventual result. We also used mixed effects models, mainly to look for factors that explain significant sources of heterogeneity within our results, but also as a part of our sensitivity analysis. Then for our results. So our overall results are depicted here in this forest plot with the black dots, including corresponding 95% confidence intervals, and sample size was given between brackets. For each of the biological processes stated here, we also plotted the results from a global meta-analysis done by Krüger et al. To see the differences. So the difference we see here when it comes to significances is that Krüger et al. Found many more significant responses, whereas we didn't find any. This could be some indication that Arctic communities are more resistant to the effects of ocean acidification, although more data will be needed to confirm this. Another interesting contrast we found is the largely negative overall mean effect size on photosynthesis, depicted here. So a possible explanation might be a decreased carbonic anhydrase and rubisco expression, which has been found to be the case in certain diatoms under acidified conditions. Being that we also included experiments done on multi-species assemblages and mesocosms, we also hypothesized that this could be due to changes in phytoplankton community structure, such as a temporarily lowered abundance of phytoplankton. However, a closer examination of both specific photosynthetic processes as well as population dynamics will be needed to confirm these. Then, as mentioned before, we also looked for sources of variability within our results. So a first source we looked at was the trophic class. So we simply compared the responses of autotrophic organisms and compared them to the responses of heterotrophic organisms. For example, for growth here, although it is not significant, we see a largely negative overall mean effect size on the growth of heterotrophs, whereas a more neutral response can be seen for autotrophic organisms. For metabolism, we can also see a difference, although once again not significant, being a largely positive mean effect size on the metabolism of heterotrophic organisms. This would indicate an increased energetic cost for heterotrophs under ocean acidification scenarios, which could probably be explained by an enhanced energy demand for the maintenance of acid-base homeostasis, which could not be compensated for by a mere reduction in growth. We also compared different taxa and their responses. Looking at growth, we found a significant increase in the growth of fleshy algae, which we used as a synonym for macroalgae. So this could potentially result from the fertilization effect, which assumes that photosynthesis is largely restricted by CO2 concentrations, and thus the increase in CO2 concentrations would enhance photosynthesis, thereby also enhancing growth. However, due to a lack of data on photosynthesis for macroalgae, we could not confirm this using our data. We also found a significant decline for the survival of fish, presented here. Although the samples included did not have any experiments on adult fish, so no general conclusions can be drawn on fish populations in general, we did find that 2 out of the 3 samples came from experiments carried out on fish eggs, and the other one from an experiment carried out on fish larvae, which does support the hypothesis that recruitment and replenishment of Arctic fish stocks could be largely impaired under ocean acidification, with potential disastrous ecological and economic consequences. We also look for sources of variability between methodical factors. The one presented here is based on the food treatment. So we simply compared the responses of organisms in experiments in which they were not fed and thus under starvation over the course of the experimental duration, compared with organism responses in experiments in which organisms were fed ad libitum and would thus not have experienced any food shortage over the course of the experiment. Although once again insignificant, we did find a visual inspection does show a difference in responses with a largely negative effect on the growth of organisms which were not fed, compared to a very neutral response in organisms which were fed. This once again stresses the importance of a proper food treatment in experiments to properly isolate the effects of ocean acidification. Now, in order to obtain a more complete picture of the effect of the environmental conditions experienced by the year 2100, we also used our data to assess the effect of elevated temperature alone, given here in the forest plot in the middle, so with the red dots. We see that elevated temperature alone leads to a significant increase in both metabolic rates and growth rates in organisms, while also inducing a significant reduction in the survival of our organisms. It should be noted that the survival data once again came only from fish eggs, but also mollusks. Once again showing the possible detrimental consequences for Arctic fish stocks, not only because of ocean acidification, but also due to elevated temperatures. Looking at the combined effects, no large effects can be observed for growth, metabolism, and photosynthesis, although this could be attributable concerning growth to the samples consisting mainly out of experiments conducted on autotrophic organisms. And as shown so far, these do tend to not show a great response to projected conditions. Survival is still largely negatively affected, although no longer significant. This brings us to the conclusions of our analysis. Although a lack of significant results, most likely resulting from an overall lack in data, did not allow us to draw major conclusions, our results did support some existing hypotheses while also providing some guidelines for future research. So first of all, our combined results provided a strong indication that Arctic heterotrophs will likely suffer the worst consequences of environmental conditions expected by 2100. However, some hope remains, as autotrophs seem to be fairly unaffected, whereas macroalgae even seem to benefit according to our data. And macroalgae have also been shown to locally increase pH values and could thus offer some refuge for future conditions for certain sedentary heterotrophic organisms. And as mentioned before, a closer examination of photosynthetic processes in the Arctic region is needed to find out exactly what was going on with the decreased photosynthetic performance that we found. And lastly, we provided a first indication that Arctic communities are possibly more resilient to ocean acidification and warming, ocean acidification alone, compared to global communities. Now that was everything. I would like to thank my supervisor for all her hard work with me. And I would like to thank all of you for your attention. Thank you.